watershed. So with that, I'm going to call the group up and let them take it away. Thanks.
Will the spacebar work to advance this?
Spacebar will work too, yep.
Okay, I'm going to do that then, if that's okay.
Why is it like
Good afternoon, everybody. We're really excited to present about our crayfish research. So, like Professor Miller said, we looked at the exoskeletons and their hardness and compared them in the Grand River watershed. I'm Hannah Shepard. I'm Julia Kloster.
And I'm Josiah Miller.
And we're going to talk to you about what we did throughout the semester. So the foundation for our research came from a concept called ocean acidification. This is a recurring problem in oceans all around the world due to an uptake in global warming and climate change. It's a chemical process that happens due to more carbon dioxide in the atmosphere. This dissolves in the water and reacts with water, and eventually it creates bicarbonate. This is a form of carbon that crustaceans and mollusks cannot use to create their protective coverings. So there are lots of studies to suggest that their exoskeletons and their shells are thinning over time. This is a really big issue because it makes them more susceptible to prey. So there's lots of issues with biodiversity because we're seeing a decline since they're being eaten more. And so we actually collaborated a lot with Professor Troy Keller in Georgia. He is doing a lot of research with crawfish right now and seeing how pH affects species diversity in certain areas and populations. So we coordinated a lot with him on how to try to apply and see if ocean acidification can be applied to freshwater environments in Michigan. So with that, we took two different streams in the Grand Rapids watershed, and we compared two streams with differing pHs due to pollution to try to mimic this ocean acidification principle.
And all that led us to our research question, which is, do the effects of ocean acidification on crustaceans with calcium carbonate exoskeletons apply similarly to freshwater environments?
So our hypothesis assumes that we can apply ocean acidification principles to freshwater environments. So we assumed that areas like Plaster Creek, which is notoriously known to be more polluted and is more acidic overall, we assumed that the exoskeleton samples we would take from this creek would be a lot weaker since we were testing the hardness of these exoskeletons in comparison to Bear Creek, which is known as Healthier Creek in the Grand River watershed. We also assumed that for all of our samples overall, we would see much harder samples and exoskeletons that had bigger claws. This is something that we just wanted to observe out of all of our samples in the entire watershed. We also wanted to make sure that people knew about issues with our pH testing. We had lots of variable pH tests. When we went to every single site, we measured pH every time, and they were very variable. They didn't really align with the fact that Plaster Creek was in fact more acidic. However, we think that this is due to when we collected our data. It was in between seasons, in between the spring and the winter months. So there was lots of snow coming and going, lots of flooding. So the influx of sediment and extra water was going through all the creeks at the same time. So we really relied heavily on past research from previous years in Bio 250 and research from our class at the beginning of the semester that did confirm that overall the pH in Plaster Creek was a lot more acidic than here.
So to get our data and our crayfish, we went to two different streams. Bear Creek, the healthy stream, and then Plaster Creek, polluted stream. We recorded the size, sex, and hardness of the cheleped, which is the large claw of the crayfish. We went to collect on all these dates, and on the 4th of April, there was an extreme flooding event, so the water was really high and really fast, so it was pretty much impossible to get any sort of data. To get our crayfish, before going out, we researched where the crayfish live in Michigan, and we found they tend to live under large rocks and in rocky areas in general. And to get them, we'd use a collection net, place it in front of a spot we had picked out, and use what we call a twist and shout method, which is getting your feet in the dirt and just upsetting all the debris that you could find. And we targeted areas that we had researched prior, and then we would systematically go upstream to make sure we are getting good results.
So once we got all of the crayfish, we brought them back to the lab at Kelvin, and we put them all in different containers and labeled them with a number so we could tell them apart. And for each crayfish, we removed the tulip head, which was advice from Professor Troy Keller, who let us know that they will regrow, and that way we didn't have to kill the crayfish to do our research. So we measured the length and width of each tulip head to avoid some confounding factors. So we decided to do a ratio of the size to hardness so that the relative size or age of the crayfish won't affect the overall mean. And then I identified the species of each one and the sex of each one, and I used the field guide to Michigan crayfish on the DNR website to do this. And we ended up only finding mainly one species, which is the northern freshwater crayfish. But yeah. And then we use this device called a durometer. This is typically used in engineering settings, but we thought that it would work for our research because we were working with comparisons and ratios. So we just needed some way to quantify hardness so that we can run statistical analysis on it. And you can see some of the different things that this device is usually testing.
So here we got pictures of the chelipeds from Plaster Creek and Bear Creek. And at Plaster Creek, they're pretty similar in size, and Bear Creek can have we have some huge ones and then some pretty tiny ones. We think that at Plaster Creek, they're all pretty similar in size because of the pollution that it doesn't let them fully grow, or that the crawfish just die before they can actually fully grow.
So for our statistics, we recorded a lot of data on each of the crawfish, but we decided to compare the hardness ratios between Bear Creek and Plaster Creek. So we made a histogram and we ran a two sample t-test, and we did get a very small p-value, which indicates that there is a statistically significant difference in means. And then we also made a scatter plot and did a correlation evaluation over both sites. So we could compare the size of the tulip head and the hardness. And our R-squared value was not close to 1, so the line doesn't fit great, but it does show the upward trend really well. And we did also have some outliers. We think that that is because there's a certain threshold that the crayfish will reach where no matter how big they get, their exoskeleton can't get any harder.
So overall, with our data, we did have statistically significant results, which was really exciting. So we decided to reject the null hypothesis and accept an alternate one. However, this alternate hypothesis is not what we projected at the beginning of the semester. So we originally thought that the crayfish samples from Plaster Creek would be a lot harder. This wasn't the case. And so what we have then confirmed in relation with our research question is that we cannot really apply ocean acidification principles to freshwater environments, but rather there's more of a relationship between size and hardness. At least that's what our data suggests. So further research would be really interesting to see if that continues.
Some sampling issues could have been a crayfish molting cycle. So we could have caught crayfish right after it had molted, which would cause the cheliped to be softer than it usually would have been. We could have completely missed some burn species because we didn't target the burn species that were found in the mud. We mainly stayed in the rocks. And it could potentially not be representative of a full population because we were trying to get just as many crayfish as we could. And like Julie said before, a durometer is not really meant for any sort of animal or like living thing. It's meant for mainly engineering use.
So like I said before, we collected a lot of data during this project, and one area specifically that we think would be interesting for some future research is the distribution of sex of the crayfish. We noticed that there were a few days, especially in Plaster Creek, where we caught mostly male crayfish and almost no females. So we hypothesize it might be because of the flooding event, or maybe the water temperature differing while the crayfish is developing. But that's definitely something that would be interesting to look into in the future.
This is a picture, by the way, of the flooding. So usually the water would have been on our knees on that day, and it was up to our chest. So it was definitely, even with all of the issues that we encountered throughout our research, such as that one, we still found results that were really interesting. So even though we weren't able to apply ocean acidification concepts to freshwater environments, there's still really interesting data that suggests that size and hardness is really important in their survival. So overall, we had a really good time finding the crayfish. We played music for them. It was a really good time. We really want to thank Professor Troy Keller. He was really foundational in our preliminary research, trying to figure out how we would collect this data. He was provided, his contact information was provided by Professor Warners, and it was really good to collaborate with him. He was equally as excited that we were doing crayfish research. Also, we did collect a lot of data, so we were thankful for Professor Miller helping us with our code and kind of just relearning that. Also, Cademan and Juman really helped us collect crayfish. This wouldn't have been possible if we didn't get enough. So that was really foundational for our research as well. We had lots of sources going through where the crayfish live and how ocean acidification can affect them. So those were some of the sources that we used. And yeah, thank you. Here's some pictures of us. Again, very excited. So yeah, if you guys have any questions, we'd be more than happy to answer them.
Thank you, folks. We have some time for questions, so 3 minutes for questions if anybody has.
Back to the slide of the, is it pronounced cheliphads?
Cheliphads.
Cheliphads, sorry. Is there any reason for the difference in color, or is that just
Yeah, it was just the lighting. We did not do a good job of standardizing that.
There were also, within the northern Clearwater species that you found, there were lots of color differences. So specifically within Plaster Creek, we found a lot of color differences. Some were blue, some were darker than others, which was really common within the species. Kind of misled us because we were excited thinking we found some new species, but they were really just color variant. That could also be something in the future that'd be interesting if pollution affects their color too. Yes.
Just clarity, what's the species name for them?
It was the
Northern Clearwater crayfish.
And do you know what the binomial
I
name is? Just
do not.
It's in the Michigan textbook.
It's in the
okay.
it's in our website.
Yeah.
The guidebook.
What other questions do you have?
So, on that box plot, it looked like you found that hardness was actually greater at Placer Creek than Bear Creek. But you also found that the crayfish at Placer Creek were generally smaller and potentially younger than at Bear Creek, which, based on your other scatter plot, would make me think maybe that they would have a lower hardness. Do you have any explanation for why that is?
Yeah, so this is something that we actually talked a lot about because we thought that the bigger claws would be a lot harder, and based on this scatter plot, it suggests that the bigger they are, the harder they will get, but we found smaller ones in Placer Creek. We think that the difference in the means, because they were so variant in size, like we found our largest and our smallest cheliped sample within Bear Creek, so we think that the difference in means definitely played out when we were doing our box plot analysis. I think that more information with water chemistry and the pollutants involved would probably help filling in those gaps as to the correlation between the two.
Go ahead. Do you have a question?
Did you find it difficult to identify male and female crayfish?
At the beginning, yes, but then once you do a few, it's pretty easy. But definitely at the beginning, I was just looking at them like, what's going on? But
Yeah, they had like little extra legs.
yeah.
Any time for one more if we have a question? All right, if not, let's give our presenters one more. Thank you.